Hello everyone, welcome to my channel. Today I'm going to talk about cell part 4, and in case you have missed part 1, 2 and 3, please check out my previous videos so that you will have continuity to what I'm teaching today. And today I'm going to talk about cell organelles of eukaryotes, continuation from the previous video. So today I'll be covering lysosomes, vacuoles, mitochondria, and plastids and chloroplasts. Okay, and even about ribosomes. And I'll be covering in part 5 about nucleus in detail. All right, so let's begin. So first, now let us talk about lysosomes. Lysosomes are also called as suicidal bags of cells. Suicidal bags of cells. These are membrane-bound structures and they are actually formed by packaging of Golgi apparatus. Right, so we all know what is Golgi apparatus. We know cis and trans phase of Golgi apparatus. So by packaging of this, these lysosomes are formed. And let me tell you something. Lysosomes are very rich in particular enzymes. You need to definitely remember these names. They all are hydrolytic enzymes. All right. Examples are hydrolases and lipases, proteases, and carbohydrases. And they are optimally at a pH which is acidic in nature. Okay, and these enzymes which are there, they are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. So lysosomes are called as suicidal bags of the cell, and they are formed by packaging of Golgi apparatus. And they have hydrolytic enzymes like hydrolases, lipases, proteases, carbohydrates, which help in digestion of carbohydrates, proteins, lipids, and nucleic acid. And they have a acidic pH. Now let us talk about vacuoles. Vacuoles look something like this. They look huge and they're membrane bound space which are found in the cytoplasm. And let me tell you something. It has a lot of important substances. It has water, it has sap, but it also has materials that are not useful. So your NCERT line says that it contains water and sap, excretory products, and other materials not useful for the cell. But let me tell you something, only contractile vacuole is the one that stores excretory product. All right. So not all the vacuoles store only waste products. They do contain important stuff also. Okay. And now if we talk about plants, like right now we're talking, this was about eukaryotes. Now let us talk about plants. What happens is the outermost layer of the vacuole is called as tonoplast. Okay. So it helps in transfer of substances. It helps in transfer of ions and materials. And this term is important for you to remember. But now coming to animals, in amoeba, where is the excretory, substance stored is nothing but it is stored in contractile vacuole. But in many cells, as in protists, food vacuoles are formed by engulfing the food particles. Now let us study about mitochondria. Now, I will tell you a brief story, okay, before I begin this mitochondria and what is the real purpose of mitochondria. So suppose you eat an apple, all right? So this apple, when you eat it, you're breaking down this complex apple into simpler substances, right? And that is converted into bolus. And I have explained this process of digestion in my digestion video. If you did not check out, please do check out. And if you have any questions, you can always ask me in the comments. So the process of breaking down of complex substances into simpler substances is called as digestion. Okay, but how do we get the energy? Like just by having a raw material that has good food is not energy, how we are consuming it, and where that breakdown of food is happening, to give energy to do work. Like, right now I'm talking to you guys, I'm explaining something, and if you're listening to me, all this requires energy, right? Even when you sleep, energy is required. And do you know who does this breakdown of energy and where it is done? It is done in mitochondria. That's the reason why mitochondria is called as powerhouse of the cell. All right. So I hope you understood why mitochondria is called powerhouse of the cell. Now, this mitochondria is not visible till it is stained properly. All right. Now, the number of mitochondria per cell actually depends on physiological activity. Now, what does that mean? Suppose if I'm moving my muscle, if I'm moving my hand a lot, there's a lot of physical activity, then there will be a lot of mitochondria over there. But if I do not, then it will be less. So basically, it directly depends on the physiological activity of the cells. Okay, and it is usually sausage shaped. So here you can see that it is sausage shaped or cylindrical, and the diameter is 0.2 to 1 micrometer, and length is 1.0 to 4.1 micrometer. Okay, and each mitochondria has two membranes. It has an outer membrane and inner membrane. Okay, see, always remember that there are two membranes in the mitochondria. It is outer membrane and inner membrane. And because of this, the inner membrane, like you can see, because of the difference between these two membranes, the inner membrane is divided again. This substance is called as matrix. Okay, so what is the inner compartment called? Inner compartment is called as matrix. All right. And in the inner compartment, you can see like how this is the outermost, but inner, you can see like these kind of enfoldings, right? Right? So that is nothing but cristae, and inside it is the matrix. So which membrane helps in the formation of cristae? It is nothing but the foldings of inner membrane. All right. And the outermost layer is the outer membrane. Okay. So I hope you have understood why it is called powerhouse of the cell, because they produce cellular energy in the form of ATP. Okay. So this was about mitochondria that you need to remember. And little bit, let us talk about the matrix. Matrix, what does the matrix really contain? It has a single circular DNA molecule, and it has few RNA molecules, and it has ribosomes, that is 70S. And these ribosomes, as we know, are mainly for protein synthesis. And how does the mitochondria divide? Mitochondria divides by fission. All right. Now, before we talk about chloroplast, let me give you a brief introduction about plastids. Plastids are found in all plant cells, and they actually impart specific colors. Okay. Don't think that, all plants are only green in color. There are plants like they are purple, yellow, and the color is mainly because of this plastids. Okay. Now, based on the type of pigments, this plastids is classified into chloroplast and chromoplast and leucoplast. All right, it is divided on the basis of pigments. Chloroplasts contain chlorophyll and carotenoid pigments. Okay, whereas chromoplasts contain fat-soluble carotenoid pigments like carotene and xanthophyllite. Mainly these impart orange and yellow color. All right. Now coming to leucoplast, these are colorless plastids with various size and shapes. All right. Now they are further divided into amyloplast, which means they store starch. An example is potato. Potato has starch, right? So you can see amyloplast in potato, and then you have elioplast, which stores oils and fats, and then you have eleuroplast, which helps in storage of proteins. So this was about plastids. All right. Now let us talk about chloroplast. Chloroplasts are lens-shaped and they are oval, spherical, discoid, and ribbon-shaped. All right. So they are lens-shaped and their length is 5 to 10 millimeters and their width is 2 to 4 millimeters. Okay. And their number varies. So it can be 1 per cell, like in Chlamydomonas, or it can be 20 to 40 per cell in mesophyll in algae. All right, so it kind of varies. So what you need to remember in chloroplast is even like how we have seen in mitochondria, they have an outer membrane and they have an inner membrane. Okay, and so it is double membrane bound. Remember, the outer membrane is more permeable, whereas inner membrane is less permeable. Okay, and inside you can see stroma over here and stacks of grana. Like grana is singular. Let me show you 1 second. Yeah. So grana is singular, but stack of grana put together is called as thylakoid. So singular is grana and grana put together is called as thylakoid. And stroma is nothing but the matrix and the connections are called as lamella. All right. So the space which is limited over here is occupied by stroma. Then thylakoid are nothing but grana that are arranged in stacks. And how they are connected? They are connected by lamella. Now this is an eukaryote and this is a prokaryote. We are going to talk about ribosomes in eukaryotes. Ribosomes can be seen in eukaryotes as well as in prokaryotes. And the first person to coin this term ribosomes is George Palade in 1953. Okay, and remember, this is a non-membranous granular structure. All right, and they consist of ribonucleic acids and proteins. Mainly ribosomes help in protein synthesis. Eukaryote ribosomes are 80S, so they are 80S, and prokaryotes is 70S. All right. Now coming to cytoskeleton. So it is nothing but you can see this is the cytoskeleton. A number of filamentous, like you see here, number of filamentous proteinaceous structure which are present in the cytoplasm is called as cytoskeleton. Okay, and what is their function? Their function is mainly for mechanical support, motility, and maintenance of shape of cells. So imagine if we do not have an outermost shape, so all our organs will be like not in proper shape. So our outer structure is determined by that. So cytoskeleton main function is determining the shape and mechanical support and motility. So let us have a review of today's class. So first we have studied about lysosomes. So let me just change the ink color so that for revision you remember. Okay. So lysosomes are nothing but they are called as suicidal bags of the cell. They are nothing but packaged molecules of Golgi apparatus, and they consist of hydrolytic enzymes, that is hydrolases, lipases, proteases, and carbohydrates, and they have an acidic pH. Okay, vacuoles store water, sap, and they also have excretory material. But the only vacuole that has the excretory material is a contractile vacuole, which can be seen in amoeba. Okay, in plants, the outermost layer is called tonoplast. Now coming to mitochondria. Mitochondria is called powerhouse of the cell because it helps in breakdown of the food to give energy. Breakdown of complex food into simpler substances is called as digestion, but breakdown of that food in order to give energy happens in mitochondria, and in the form of energy is released in the form of ATP, adenosine triphosphate. That is the reason it is called as powerhouse of the cell, because cellular energy is in the form of ATP. Okay, it is not visible until it is stained properly. And number of mitochondria depends on physiological activity of the cell. It is double membranous structure. It has an outer membrane and inner membrane. Inner membrane has like inner foldings divided into cristae, which has matrix, and outer membrane is the outermost layer. And the matrix has a single circular DNA and an RNA molecule, and it has ribosomes that is helpful for protein synthesis. Okay. Now coming to the chloroplast. Chloroplast is also double membranous structure. It has an outer membrane and an inner membrane. Outer membrane is more permeable as compared to inner membrane. And you can see grana, each one is called as grana, and they are stacked up and they form thylakoid. Okay, their length is 5 to 10 millimeter, width is 2 to 4 millimeter, and number varies. Like in chlamydomonas, one chloroplast can be seen by cell, whereas in algae 20 to 40 per cell can be seen in the mesophyll. And like chloroplast is nothing but a type of plastid. So plastids mainly give specific colors. They are divided based on type of pigments. So chloroplast has chlorophyll, which is a carotenoid pigment. Chromoplast is having fat-soluble carotenoid pigments, that is carotene and xanthophyllate, that imparts a yellow and an orange color. Leucoplast is colorless plastid with various size and shapes. It has amyloplast, which you can see in potato in the form of starch, elaioplast in the form of oils and fats, and eleuroplast in the form of proteins. So, and now coming to ribosomes, you can see 80S ribosomes in eukaryotes and 70S in prokaryotes. And ribosomes' main function is nothing but it helps in protein synthesis. And ribosomes was first discovered by George Palade in 1953. Cytoskeleton is nothing but the number of filamentous proteinaceous structures that can be seen in the cytoplasm. That is, it is nothing but this microfilaments, which are like this, and microtubule. These are all, like, we'll be talking about it in detail, but remember, number of filamentous proteinaceous structure that are present in cytoplasm is nothing but cytoskeleton, and the main function is it helps in mechanical support, motility, and maintenance of shape of the cell. So thanks for watching my channel, and in tomorrow's class, I'll be covering cilia. So tomorrow's class will include cilia and flagella, and centrosomes and centrioles, and nucleus, and microbodies. All right. So in case you guys have any questions, please leave a comment and I will reply to it. And also do not forget to subscribe so that you will receive notifications whenever I release a new video. And in case anyone of you want the PDF notes of this class, I'll be very happy to send you. But then for that, you need to leave a comment saying that you would be needing PDF notes for a specific topic. All right. So thanks for watching my channel and stay tuned to my channel. And in case you have not yet subscribed, please do subscribe. Thank you.